So my interest in this disease started a few years ago when we wanted to test the idea of using gene therapy to transfer a specific gene to the liver cells in the mouse model of alpha-1 antitrypsin deficiency to see whether the gene transfer of this particular gene was able to clear the accumulation of mutant alpha-1 antitrypsin. So the gene that we wanted to test is called TFAB, and that's a transcription factor that was, at that time, recently discovered to be involved in autophagy. So autophagy is a process that is present in every cell, and it's basically the ability of cells to self-digest their damaged material. Actually, the work of Jeff a few years back showed that autophagy is involved in the disease. So then we thought, well, if we transfer this master regulator gene of autophagy, we could activate this process in the liver cells, and we can get rid of this mutant alpha-1 antitrypsin. So actually, the results that we had were quite striking and surprising, because you have seen from the picture that Jeff showed that these liver cells accumulate this pink material that is the polymerized alpha-1 antitrypsin. And following the injection of our gene therapy vector encoding this TFAB transcription factor, we could see really quite marked and dramatic reduction in the amount of the polymer that accumulated. So basically, we showed that if we and this is consistent with previous observation made by Jeff, that stimulating this process that is present in the cells, that is called autophagy, we can get rid of this toxic protein that accumulates in the liver. So transferring a transcription factor obviously may not be the safest option, but because these transcription factors, as many other transcription factors, do many things, does many different things. And so ideally, well, the experiment was for us was very informative, because it showed clearly that stimulating autophagy, it's a potential target for to eliminate, to clear the accumulation of alpha-1 antitrypsin in the liver. But ideally, we would like to do the same thing by activating pharmacologically, maybe, this transcription factor, and possibly by pulsatile treatments that can activate temporarily the transcription factor to get rid of the excessive polymerized alpha-1 antitrypsin. And this is what we are trying to do. We are also trying to understand how other pathways that are altered in the livers could impact on the activation of autophagy, or could be modulated to eliminate the excessive toxic alpha-1 antitrypsin.